Okay, let's look at the factors that affect androgenesis. Okay, so they are, the first one is the genotype of the donor plant. Okay, so means the genes or the chromosomes inside. So obviously, if you want to get a successful culture, no, a successful plant, a successful production of your hybrid of your plant that you require, the genes have to be good. Okay, good means healthy, not resistant, resistant to diseases. Okay, or maybe suppose you need higher yield capacity, right? Or if you need uh, suppose you need the leaves, then a higher shoot producing capacity. Or if it is a a tuber forming plant, you want the buds, okay, or the spuds, then you will choose a root forming capacity plant. Okay, or if you want a disease resistant one, very highly diseased, very disease place where there's a lot of disease, you want a disease resistant. Or where there's a place you want to plant it, you want to send these plants into a very dry area, you want drought resistant. Okay, so you have to choose a good genotype of your donor plant. Okay, parent, I don't exactly mean parent, means initial plant. Okay, because there's no fertilization occurring here, we are just directly planting in a fake environment, no. Then not only that, the way you breed, the way you grow it also, you have to get a good technique for it. Okay, the next is stage of the microspore or the pollen. Okay, you have to select the anthers at the right stage of their pollen development. Okay, it could be from the tetrad to the binucleate stage. Okay, whichever it suits your experiment. Then anthers at a very young age and a late stage are not suitable. Okay, too late also not good, too old also not good. Okay, so this all depends on the species. Okay, then you have to look at the status of the donor plant. So they grow under the best natural environmental conditions with good anthers and good pollen. Okay, the flowers obtained from young plant at the beginning of the flowering season are highly responsive. The use of pesticides should be avoided at least 3 to 4 weeks before sampling. Okay, when we say physiological status of the donor plant, it means that before you take the anther, you take the pollen, before you do the androgenesis experiment, okay, you observe that donor plant, you observe it. Okay, you observe, is it living in a good environment condition? Does it have pesticides? Okay, is it healthy? Okay, so these are the things. Like over there it's already written, you should grow the donor plant in the best environment condition and do not use pesticides. All right, then next you do the pre-treatment of the anthers. So you have to treat the anthers so that we stop the conversion of the pollen cell into the gamete. Okay, so we don't want the gamete or the sperm cell to form. So we have to stop it at the stage that we want. We don't want the tube nucleus and the sperm cell. We don't want that. We want before that. So you have to stop the stop the meiosis too. Okay, then what's there? So we could use treatments by chemicals, by temperature. Okay, so certain chemicals, they can induce parthenogenesis. I told you, parthenogenesis, directly a child will form from a gamete without fusion. Okay, so there are some chemicals like 2-chloroethyl phosphonic acid, or the short form name is ethyl So when we treat the plants with ethereal, multinucleated pollens are produced. Okay, and if we culture these pollens, we will get embryos. Okay, then you see the temperature. So if you treat them with cold temperature for about 3 days, induction occurs to yield the pollen embryo. Okay, normally, okay, you don't do any chemical, you just take flower bud and you keep in the cold room, okay, for 3 days, and then you will find pollen embryos. So example is datura and the nicotiana. Again, even better androgenesis if we store these anthers in the low temperature. But opposite to low temperature, you can also use high temperature. There are some species like mustard and capsicum where you give high temperature like 35 degrees Celsius, it will give you the androgenesis. Okay. Then the effect of light. Haploids are produced better when there is light, but there are some plants that grow well in both the light and the dark. Okay. If we isolate only a pollen, not the anther, the pollen is sensitive to light. Okay. Then let's look at the culture medium. I've already given you a little bit, some idea you already have, because I did with you this plant tissue culture, no? So to have a successful culture, anther culture, you have to you have to design your media in such a way that you get exactly what you want. Okay, you get healthy haploid plants. All right, but there is no such universal medium which is perfect. Let's say my son, he drink lactogen, then my friend's son, he drink nans, then my other friend's son drink pediasure. If I give my son pediasure, he doesn't like. If my friend's son take lactogen, he don't like. So just like babies, they select the food, no? Some of you also, when you grew up, you like beans but you hate carrots. Then another of you, you like lots of chili but you hate potato. Certain things like this, no? So even plants, different plants require different composition of the media. Some plant require more nitrogen in their media, some plant require less. Some plant require more phosphate, some plant require less. Some plants require both high amount of phosphate and nitrate and sulfate. Some plant require very less. I think you get it, okay? So commonly used media for anther culture are the MS media, the White's media, the niche media, the N6 and the B5 media. Okay, these are different names of media. So you write MS media like this. Okay, or White's media like that. Okay, just one or two, you can remember enough. Sucrose, nitrate, there, this one I'm reading, this paragraph. Sucrose, nitrate, ammonium salts, ammonium acids, minerals are essential for androgenesis. So just like your normal plant, when you grow at home, you require macronutrients and micronutrients, same. And then you require growth regulators like auxins to produce roots and cytokinins to produce shoots. Okay. So addition of glutathione and ascorbic acid, these are vitamins, okay? Glutathione and ascorbic acid promote angiogenesis. So even you and me, we require vitamins, isn't it? So same here, plants also, they require these vitamins. So when you add that culture medium with activated charcoal, they even grow faster because charcoal has carbon, no? So they grow even faster. It's because the charcoal removes the inhibitors from the medium and facilitate haploid formation. Okay.